Hello everyone, my name is Hubert and welcome to Teach Me channel. Today we will be learning about A-level prokaryotes and viruses. Previously we covered eukaryotes. We covered cells such as animal cells, plant cells and fungi, which are all types of eukaryotic cell. Now, the other type of cell are prokaryotes. One very good example of prokaryote is bacterium, or bacteria in plural. Now, if you haven't watched the eukaryote video, if you're not confident on what components, what structures, and what the functions of each structures of eukaryotic cells are, well, check out the link in the description below. I go over all the basics of eukaryotic cells. So just have a watch and then you can come back here and you'll be much better off in this video. Okay, without further ado, let's get started. Okay, so as I said, one example of prokaryotic cell are bacteria, or bacterium is the singular form of that. Now, prokaryotic cells are much simpler than eukaryotic cells. What goes with that is that bacteria are much smaller than normal animal cells. I think, what was it, about 100 bacteria can fit into one animal cell, if I am not mistaken. Now, anyway, the point is that they are much smaller. And their structures differ slightly to that of a eukaryote. Okay, so let's get started. So, first of all, we have a plasma membrane. It is consisting of proteins and lipids, so very similar to eukaryotic cells. And again, in this case, plasma membrane's function is to, first of all, keep all the things that are supposed to stay inside bacteria, keep all these organelles inside the bacteria inside, and to keep everything else outside. And also plasma membrane controls what gets in from outside and what gets out from inside. So that it's very similar to eukaryotes. That's the function of plasma membrane. Now, moving on, we have ribosomes. Again, the function of ribosomes in bacteria, just like in eukaryotic cells, is to make protein. Ribosomes in bacteria also make protein. However, the difference here is that ribosomes are much smaller. Bacterial ribosomes are much smaller than eukaryotic ribosomes. That being said, as I said, their function is the same. Okay. Next one up is cytoplasm. Now, cytoplasm in a prokaryote is, it contains all these enzymes and all these compounds required for chemical reactions within that our little bacteria. What is the difference? Well, first of all, these compounds themselves are likely to differ. Bacteria is likely to have different enzymes in cytoplasm than, say, eukaryotic cell. So that might be the first difference, but another important difference is that bacteria has no membrane-bound organelles. Now, what do I mean by that? Well, eukaryotic cells, for example, have many organelles that are bound by membrane. I mean, one such example is mitochondria. You have mitochondria which has mitochondrial membrane. Plant cells, also on top of mitochondria, plant cells will also have chloroplast. Again, that is membrane-bound organelle. Now, important thing here is that bacteria has no such membrane-bound organelles. Ribosomes are not membrane-bound, so that's the difference. Okay. Often bacteria, not all bacteria, but often bacteria can have flagella. Flagella help them move around. That's their main function. Flagella helps bacteria move around. And so obviously that is important if a bacteria wants to invade a host organism, or if it even wants to just move to capture some resources from outside. So so there are many different patterns of flagella. Some bacteria can have flagella all around them. Some bacteria can have multiple flagella at the back. Some bacteria have single flagella at the back. Some may have none. Some bacteria may have no flagella. Okay, here's the big difference. Here we have a circular DNA. So this is essentially a coiled strand of DNA. This is bacterial sort of core coiled strand of DNA. Now, what's the difference? Well, I mean, as you probably know, the core concept of eukaryotic cell is that it has a nucleus. Well, here we have no such nucleus. We absolutely have no nucleus. We just have freely floating strand of DNA. It is not captured within certain membrane. It is not captured within certain structure. So that's a major difference between eukaryotic organism and prokaryotic organism. Now, here we have plasmid. So what are plasmids? Plasmids are very small circular strands of DNA. They are separate from the circular DNA. They also are circular, but they are separate from circular DNA. And bacteria can have one or more plasmids within its cytoplasm. Now, these little circular strands are important because they can be transferred between bacteria. So this bacteria could give this plasmid to another bacteria. And these plasmids can contain many, many genes. So each plasmid can contain different DNA. So it can code for different things. So, I mean, significance of that to us is actually that bacteria can share, they can transfer their antibiotic resistance across to another bacteria using plasmids. So they can have a antibiotic resistance gene on this plasmid, say somewhere here, and then they can give that to another bacteria, and then that another bacteria becomes also antibiotic resistant. It starts producing these enzymes that can break down these antibiotics before they can do any real harm to the bacteria. So these little guys, these little structures are very, very important. Okay. Moving on, we have cell wall. So much like in a plant cell, bacteria have cell wall. And if you think about it, again, bacteria have to swim around in many different environments, human body, outside human body. So they will be subjected to many differing concentrations of solutes. So therefore they will be subjected to many different concentrations of water. So this cell wall, again, helps them cope with it. It helps them intake water without bursting. It helps them lose water without becoming too crenated. Now, so that's very important. However, the difference here is that in a plant cell wall is made from cellulose. Now, in bacteria, the cell wall is made from glycoprotein mostly. It's made from glycoprotein. So they are proteins with a sort of carbohydrate or sugars attached to them. That's what glycoprotein is. And they use those glycoproteins to signal to each other. So bacteria can signal to each other using that cell wall. So that's sort of another smaller function of that cell wall. Okay. And the last but not least, some bacteria may have capsule. Capsules are very diverse. They can be made out of very different compounds, glycoproteins, some proteins. It's a mix of different substances. And capsules help bacteria stick together. Cell wall also helps them stick together. So cell wall and capsule they can help bacteria stick together in groups and form actual colonies. And also capsule, more importantly, capsule is very, very important in immune system avoidance. The capsule may contain various proteins that help it avoid immune system. And the capsule also covers these little glycoproteins in the cell wall, which often are used by immune system to detect that this in fact is a bacteria. So if the capsule covers those little glycoproteins, then immune system essentially can't see a bacteria. So again, to just sort of summarize, capsule is important for immune avoidance of immune system. Okay, so this is our bacteria, and bacteria, this is our sort of good example of a prokaryotic organism. As you can see, there are some major differences. There are some similarities, like plasma membrane, there are ribosomes, there is cytoplasm, but there are some major differences. I mean, flagella can be in prokaryotic organisms. It is very rare in eukaryotic organisms. There are actually cells within our body that have flagella, but it's much rarer than in bacteria. There is a circular DNA as opposed to nucleus. There are plasmids. There are no plasmids in eukaryotes. Cell wall made from glycoprotein, that's different to plants. And capsule, which is not a thing in eukaryotic organisms. Okay, so that's it for prokaryotes. Now let's move on to viruses. One second. Here we go. Okay, so that's our little virus. Viruses are small. Viruses are even smaller than bacteria. So they are very, very small. They are very small. Many viruses actually fit inside bacteria. Now, important thing to consider is that virus is not a cell. Bacteria is essentially a type of a cell. Eukaryotic cells are, well, they are eukaryotic cells. Virus, however, is not a cell. It is just some protein with nucleic acid. But absolutely, it is not a cell. Okay. And also, viruses, this is debatable, but currently it is believed that viruses are not a form of life. I know this is confusing, but there are certain processes that constitute as being life. And virus doesn't have those processes. So, virus doesn't have, for example, any tangible form of excretion as many other cells do and many other life forms do. So virus currently is not considered a life form, which is debatable at the moment, but at the moment is not considered a life form. Okay. So also, how do viruses reproduce? We sort of touched upon that in one of the previous videos, if I remember correctly. Viruses cannot reproduce by themselves. This, by the way, is another reason why some people don't believe viruses are alive. They can only reproduce by hijacking another cell, another bacteria's mechanism. So what do? They attach to a bacteria or a cell, and they inject their DNA or RNA, as we'll go over in a minute. They inject their genetic material, which fuses with the genetic material of a bacteria or genetic material of a cell, human cell. And because that genetic material fuses, it basically is being used by the cell. So the cell essentially unwittingly starts creating more and more of these viruses that are encoded by this genetic material. And those viruses essentially can leave the cell and go on to invade another cell. So that's how viruses reproduce. Now, okay. So now let's move on to what is what in a virus. Okay. First of all, we have a capsid. Capsid is a protein sort of binding. It's a protein capsule. In other words, it's sort of like a protein ball. And it holds the genetic material inside. So it holds what's supposed to be inside, and it gives that virus a bit of a structure. The capsid allows it holds the attachment proteins. Which are proteins that are necessary to attach to cells. This is important in virus invasion. It uses these attachment proteins to attach to cell or a bacteria and to commence the whole invasion sort of process. And last but not least, we have genetic material. So we have a genetic material inside the virus that will be injected into the cell or a bacterium and will fuse with it to produce more of the virus. Now, that genetic material is, as you can see, there is no nucleus, no such a thing, or anything like that. It is sort of free-flowing genetic material inside that capsid. And that genetic material, unlike eukaryotic cells and prokaryotic cells, that genetic material can be RNA. DNA is what eukaryotic cells have and what prokaryotic cells have. It's a double-stranded, genetic molecule, nucleic acids. They're sort of double-stranded and form bases, whereas RNA is single-stranded. Now, so some viruses can have RNA. Some still do have DNA, but some can have RNA. And the viruses that have RNA are called retroviruses. They're called retroviruses. Okay, so this is it for today, guys. As ever, thank you very much for watching. I really appreciate this. And if you want to stay up to date on daily science, maths, and some biomedical science videos, hit that subscribe button below. If you like this video, if you think it helped you, give it a big thumbs up. And again, thank you very much for watching. If you have any questions, just shoot me a question in comments. I'm more than happy to answer to any queries you may have. And see you next time.